Hello, everyone. Thank you for joining us today for our webinar titled Navigating Preservative Testing, Ensuring Safety, Quality, and Compliance. So this webinar will feature presenters Amanda Bird and Dr. Fei Wang, experts from two of Eurofins' U.S. food custom testing laboratories. All right, so first, I would like to provide a little background on our presenters for context of their presentation today. We have two presenters, one being Amanda Bird. She is the site manager of Eurofins SF Analytical in Horsham, Pennsylvania, about 30 miles outside of Philadelphia. So Amanda has been with the GMP microbiology team for 15 years, providing microbiological testing support for pharmaceutical, OTC, and personal care companies, including preservative efficacy testing of products across those various industries. Amanda's special service laboratory is registered with the FDA for GMP compliance, is ISO 17025 accredited for microbiological USP testing and also infant formula testing, and is registered with the DEA to test Schedule 1 through 5 controlled substances. So welcome, Amanda. Our other presenter, Dr. Fei Wang, is a senior research microbiologist within the Eurofins Advanced Food Microbiology Group. So this experienced team based across the country in Fresno, California, operates a biosafety level 2 laboratory focused on microbiological research projects, regulatory compliance, and process authority support. So Fei has been with the Advanced Food Micro team for over 6 years, providing microbiological consulting for food and biotech companies. Fei specializes in preservatives and antimicrobial agents in food, nutritional supplements, and healthcare products. She holds a medical degree from China, as well as a master's and PhD in microbiology from Iowa State University. All right, with that introduction, I'm going to go ahead and hand it over to Dr. Wang to begin.
Thank you for Scott's introduction. Let's start with the overview of today's presentation. First, we will talk about how preservative testing begins in R&D and early stage development. Next, Amanda will discuss the final product testing and meet regulatory compliance. Finally, we will discuss to bridge two together with an example case study. Now let's talk about part one. Preservative testing in R&D and early stage development. Why do we need preservatives? Preservatives do two big jobs: keep the product safe and make them last longer. From the product stability and shelf life side, preservatives play a critical role in preventing spoilage caused by the organisms like lactic acid bacteria, yeast, and mold. By inhibiting the growth of those organisms, preservatives help keep the food to keep them fresh, maintain the texture and flavor, and also help to extend the shelf life.
In terms of pharma and nutritional supplements, preservatives also play a critical role in preventing microbial growth, which helps extend the shelf life of the product and ensuring that the product remains effective for a longer period of time. These preservatives serve as a key ingredient in contributing to the overall stability of the product by preventing degradation and maintaining the integrity of the active ingredients. When looking at personal care products, the role of preservatives in shelf life is similar to that in pharmaceuticals, where the preservatives allow these products to remain safe and effective longer by preventing microbial growth. These preservatives also help maintain the quality and appearance and effectiveness of the product. Without them, products might change in color, texture, and even smell.
From the safety and the compliance side, preservatives play an important role in inhibiting the growth and reducing the pathogenic organism, which can cause serious illness. For example, like E. coli, Listeria monocytogenes, Salmonella, and so on. The preservatives is very important when the food product relying on their formulation alone to inhibit or kill the pathogens to meet FDA or other regulations?
Most types of pharma and personal care products are packaged as multi-dose formulations. Preservatives are essential to prevent microbial contamination from repeated use and keeping consumers safe from potential infections and skin irritations. These products are tested using USP 51 to ensure the preservative is in compliance with regulatory standards.
The preservatives can be broadly categorized into artificial and nature types. Nature preservatives including vinegar, which is widely used in dressing products, and plant extracts like rosemary extract and microbial extracts such as niacin, and pure secondary metabolites like fiber from mushrooms, and essential oil such as tea tree oil. They are very popular to be used into organic, house food, and nature food with clean label. And and artificial preservatives such as sodium benzoate and potassium sorbate that are commonly used in processed foods, beverages, and baked good products.
Many personal care product manufacturers are now using more of these natural preservatives, the essential oils, such as tea tree oil, clove oil, and thyme oil, to try to keep the products more natural. They're moving away from the use of artificial preservatives like parabens due to concern arising in potential toxicity. Pharmaceutical manufacturers will utilize more of these artificial preservatives like sodium benzoate and potassium sorbate to formulate oral solutions, and benzalkonium chloride is typically used in topical medications and nasal sprays.
For preservative testing in R&D phase, we have several testing methods for efficiency of the preservatives. Those including the agar diffusion method, broth dilution method, time kill test, and challenging studies. Each method has its own set of advantages and is choosing based on the specific requirement of the study. Please note that agar diffusion and broth dilution method are growth study since they can only detect if the growth of the organism can be inhibited or not. Time kill test is a dying off study since it's used to evaluate if the organism could be killed. And challenging study can be both, can be tested for growth and dying off. Let's take a closer look at each method with case studies. In our first case study for agar diffusion method, which is a simple and effective way to screen preservatives for their ability to inhibit microbial growth. There are two primary techniques used in this method: agar disc diffusion method and agar wheel diffusion method. The agar disc diffusion method shown in the left picture, small paper discs soaked with the preservative solutions are placed on the surface of the agar plates, which have been inoculated with target organisms. If the preservative has the antimicrobial effect, you will see a clear zone around the disc where the microorganism cannot grow. This zone of inhibition shows the preservative can inhibit the growth of the target organism. On the right side, we have a case study using the agar well diffusion method. The wells are drilled in the agar and the preservative products are added directly into the wells. This method is particularly used for when testing larger volumes of the product or when the product is not easy being observed by paper discs. In this case study, we tested two preservative products against Staphylococcus aureus. The results show that the preservative product number 2 had promising activity as evidenced by the clear zone of inhibition around the wheel, which is circled in red in the picture. However, preservative product number 1 didn't show any antimicrobial activity around the wheel. Next one. The agar diffusion method is a great initial screening tool because it is simple, cost-effective, and provides visual and easy to interpret results. However, it is not suitable for preservatives that cannot diffuse through the agar, and also quantitative results often require further analysis. The next method is broth dilution method, which is widely used to test the effectiveness of liquid preservatives. The process involves diluting the preservatives with different concentrations in buffer or broth solution, then inoculating each dilution with the target microorganisms. After incubation, microbial growth is evaluated by checking for liquid turbidity or use a color indicator. This method helps us identify the minimal inhibit concentration, which we also call MIC, for the preservative against target organism. In our lab, we use a spectrophotometer equipment to monitor microbial growth in real time. The spectrophotometer equipment can measure the optical density, we also call OD value, of the liquid, which reflects the growth of microorganisms. By recording OD value at regular intervals, we were able to generate a detailed growth curve that show how the microorganisms grow under different preservatives. In this case study, we tested 5 preservatives against Listeria monocytogenes. The x-axis represents incubation time, and the y-axis represents OD value. Higher OD values means more turbidity, which indicates more microbial growth. The results show that product 2 and product 4, which represent by orange and the yellow lines, has the lowest OD values, means they were the most effective at inhibiting the growth of Listeria monocytogenes. The advantage of this method is it can provide detailed growth curve, giving us a deeper understanding of how the preservative work. Compared to traditional method, it is more efficiency and can give us more comprehensive data. However, there are also some limitations. It can only be used with clear liquid, and also spectrophotometer cannot distinguish between live and dead cells. So we can only detect the growth, but we cannot access the reduction of the microorganisms. The third method, time kill test method. It is the method to be used to access the reduction of the organisms caused by the preservatives over time. By incubating the target organism with the preservative and sampling at different time interval, we can determine the numbers of the viable cells by plating and counting the colonies. This method is particularly useful for quantifying the reduction of target organism caused by the preservatives. It helps us to see how effectively the preservatives kill the organism, so we also call it die-off study. In this case study, we use the time kill test to access the reduction of Salmonella subspecies and Candida albicans at different preservative concentrations. The x-axis represents incubation time, and the y-axis shows the survival of the target organisms. By plating inoculated sample at each time point, we observed the clear decline for both Salmonella and Candida albicans at some levels. In the left side, for the Salmonella, we can see the reduction of Salmonella at level 1 and level 2 concentration, and the level 1 works better, means level 1 is most effective. On the right side, we observe the reduction of Candida albicans at level 1 only. So only this concentration of preservative is working to inhibit candida albicans at level 1. This method can help us understand how effectively the preservative reduce the target organism, but it is relatively expensive and time-consuming. The last method is challenging study. They are designed to evaluate the real-world effectiveness of preservative in actual food or nutritional supplement products. Those studies involve formulating the product with the preservatives and then inoculating with the target organism and plating at each sampling time to evaluate the microbial growth or inhibition. Challenging studies provide realistic evaluation of the preservative's performance under practical conditions. They offer very valuable insights into how the preservative will perform in the actual final product. In our final case study, we conduct a challenging study to evaluate the fate of yeast in different products formulated with the same preservative. While the preservative's efficiency was initially tested in a screening study, when formulated in actual food products with different pH, water activity, and Brix, still can show with different results. In this case study, we observed product 1 and product 3, which is green and the blue line, didn't support the yeast growth in those two products. However, product 2 and product 4 can support the yeast growth. Those challenging studies in actual products are essential for ensuring that the preservative also provide the expected inhibition under the influence by the formulation or the manufacture processing of the actual products.
Okay, now we will dive into part 2 of the presentation. In this section, I will discuss preservative testing in final products by use of USP 51 analysis and how analysis under a GMP quality system demonstrates the product's compliance with the regulatory standard. I will now get into more detail on USP 51 antimicrobial effectiveness testing. This is also commonly referred to as preservative efficacy testing or preservative challenge studies. USP 51 can be applied to any aqueous-based product. There are 4 standard categories. Category 1 includes injectables, otic, and sterile nasal products. Category 2 includes topical used products. Category 3 includes oral products other than antacids. And category 4 includes antacids that are made within aqueous space. This analysis is performed using 5 standard microorganisms, which include gram-negative bacteria, gram-positive bacteria, a yeast, and a mold. Specifically, E. coli, Pseudomonas aeruginosa, Staph aureus, Candida albicans, and Aspergillus brasiliensis. The principle of the method is to determine reduction of microorganisms over a 28-day period. After the samples are divided into equal aliquots, the aliquots are inoculated with bacterial and fungal suspensions, which are added to the product at a level between 100,000 and 1 million colony-forming units per mil of product. The inoculated product is incubated at 20 to 25 C until sample aliquots are pulled for testing. At specific time intervals, which is dictated according to the category type, the sampled aliquots are serially diluted in broth. The number of viable cells present is determined by plating an agar and counting the colonies. The log 10 value is then calculated, and the change in concentration is expressed in terms of log reductions. The preservative effectiveness is determined by comparing the calculated log reductions to the acceptance criteria. I will now get into reporting and determining compliance with the acceptance criteria. On the left side of the slide, is a snapshot of the acceptance criteria by category type. Without getting into too much detail on each of the acceptance criteria per category, I will just provide a brief overview. With the exception of category 4, all bacterial organisms must meet a minimum log reduction. For category 4, the bacterial organisms cannot have an increase from the initial calculated count. For all 4 of the categories, the acceptance criteria for yeast and mold is that there must be no increase at each test interval from the initial calculated count. No increase is defined as not more than a 0.5 log 10 more than the value to which it's being compared. So essentially the number of CFUs can increase and still meet acceptance criteria, as long as the log 10 value is not greater than 0.5. On the right side of the slide is an example report that is provided by Eurofins. Our report will provide the initial inoculum concentration, total number of CFUs recovered at each test point, and then the log reduction at each of these test points to make it easy to determine if the product is in compliance. Now getting into the regulatory side. Testing performed under good manufacturing practices follows a quality management system that ensures products are tested according to rigorous standards and meet specific requirements for safety, quality, and efficacy. Detailed written procedures and documented proof of adherence to those procedures are essential for demonstrating GMP compliance at each step of the testing process. Our documentation records and practices follow the ALCOA principle, which is a set of principles established by the FDA to ensure data integrity in records. Per this principle, all records must be attributable, legible, contemporaneous, original, and accurate.
Okay, let's move to the part 3, bridging the R&D and the final product testing. How to integrate R&D and final product testing. In the R&D phase, our focus is on screening the preservatives and conducting preliminary efficiency tests to identify promising preservatives and concentration. We can test many different types of preservatives against the target organism to determine their potential antimicrobial activity. We select the test methods like agar diffusion method, broth dilution method, and time-kill test, depending on the preservative types, target organisms, and client objectives, to evaluate how effectively a preservative inhibits or kills the microorganisms. Those early stage tests help us to narrow down the most effective options before moving to the final product testing.
So once the project has been transferred to the laboratory for final product testing, the sample is set up to undergo USP 51 analysis. The samples are analyzed under GMP conditions, and the results will be evaluated to see if the product is compliant with the regulatory standard. This integration provides a seamless partnership and ensures products are effective, safe, and market ready.
And we always customize the study for our clients. The key is to design the studies that align with the specific product, manufacturing, processing, and the client objectives. For the food product, we focus on the real-world condition. When we design the study, we consider the factors like the product formulation. It is low acid or high acid food. It is low or high water activity food. It is liquid or solid. We also consider the manufacturing process. Is it thermal processed or non-thermal processed, like high pressure processing? Or does it rely on the formulation itself? Additionally, we design the study to meet the client's objective. Are they looking for extending the shelf life or achieving a specific log reduction of the organisms? or they want to meet any regulatory requirements, like the 21 CFR 114 from FDA requires 5-log reduction for pathogens in acidified food, or any other regulations from USDA or EFSA in Europe. By considering all those factors, we design studies that are not only scientifically sound good, but also practical and aligned with client needs.
When looking at the preservative selection in pharmaceuticals and personal care products, there are several factors that must be considered. A few of these include route of admission, who the consumer will be, compatibility with other ingredients, and effectiveness against specific target microorganisms. For example, sodium benzoate and potassium sorbate are commonly used in oral aqueous preparations due to ease of solubility. Benzalkonium chloride is commonly used in topical preparations over oral preparations due to it giving a bitter taste. And benzoate derivatives are not recommended for babies due to their inability to metabolize and eliminate benzoic acid. While these products will require USP testing, which is specific in terms of claiming product compliance, these studies can also be customized to meet client needs. In some cases, clients may have a specific target microorganism they would like to have challenged. These situations typically arise when a particular bug has been repeatedly found in a water system or during environmental monitoring of the manufacturing area. In these cases, these microorganisms can be added to the testing as special requests.
Let's look at an example case to show how we design preservative testing for personal care product from R&D phase to final product phase. In this case, the goal is to develop an essential oil-based preservative for personal care products. We want to keep the product quality and also ensure the antimicrobial effects for common spoilage organisms and pathogens. To do this, we select some common spoilage organisms like Aspergillus ligers, Candidus albicans, as well as some pathogens like Staphylococcus aureus and Streptococcus mutans, which are common skin and oral pathogens. We also conduct a literature review to choose essential oils based on their potential antimicrobial activity for selected target organism, such as tea tree oil, cinnamon oil, rosemary oil, and lavender oil, and test them at different concentrations. The study was designed to meet regulatory standards like USP 51 and also including some challenging tests to evaluate how well this preservative worked in the final product. First, we use the agar diffusion method to screen the antimicrobial activity of selected essential oil. This helps us to narrow down which oils are the most promising preservative against target organism. Next, we apply the broth dilution method to determine the MIC of each essential oil. This step allowed us to identify the most effective concentration for future testing. Finally, we used the time kill method to evaluate the reduction of essential oil against the pathogens. Based on those screening studies, we identified the most effective essential oils and the optimal concentration for the next step, USP 51 testing.
Once the essential oil and concentration was identified, the sample was transferred to the GMP microlab for USP analysis. The results at 14 and 28 days were compared to Category 2 acceptance criteria. The product successfully met the requirements.
To wrap up, let's recap some key takeaways from today's presentation. First, the preservative testing is essential. It keeps the product stability, safety, and compliance with industry regulations. Without the proper testing, there are serious risks of product recalls and even potential harm to the customers. Second, the journey from R&D to final testing is critical. R&D phase can screen preservatives to identify promising preservatives and the optimal concentration regarding to the target organism. And the final product testing follows the regulations like USP 51 GMP challenge test. This is where science meets regulations, making sure the product works in the real world condition. And finally, customized solution. Whether it is food or pharmaceuticals or personal care products, each industry has unique needs based on the product type, target organism, and regulations. That is why customized study is the key. Working with the experts ensure your product is safe, effective, and ready for the market success.
This concludes our presentation. We would like to thank you for joining us, and we're going to kick this back over to Scott for any questions.
Great, thank you so much, Amanda, Faye. We did have a few questions come in. I'll get through them. All right, so I'm going to go ahead and start with a difference in artificial and natural preservatives. So question being, if a company wants to switch from artificial to natural preservatives, what should they do? What considerations should they make?
Yeah, when the company want to switch from artificial to nature preservatives, because nature preservatives now is more and more popular in the market because the clients the customer looking for more nature and clean label product. First, we need to understand the final product type. What is the product? It is a food or pharmaceutical product, or it is a personal care product. And also we want to understand the purpose of the preservatives. They use artificial preservatives to extend the shelf life or relying on to get some log reduction. When they want to change to the nature preservatives, or they want to just inhibit the spoilage organism or replace the like manufacturer processing, like thermal processing. So based on those different objectives, we have to think about the type of the nature preservatives. Also, looking for when regarding to the types of the preservatives, if you have some preservatives on the market in mind, then we can double check it if they are working in your product type and also working against the target organism. And also we can do some literature research because there are so many literatures talking about the nature preservatives and what they are working range, like concentration and working environment in acid or neutral product, and also talking about the target organisms. And another way is we can also talk about the preservative manufacturers because they typically have their own data to show how their product performing. So based on those data, literature search, and the manufacturer internal data, we can narrow down the list of the nature preservatives, try to pick up some candidates of nature preservatives, and also at least have a range they are working. If there is no range, it's also fine. We can do some screening study. And then to the next step, based on those so many preservatives, we can do the screening test as what I presented in the presentation. We can do the agar diffusion method, broth dilution method, and time kill test, depending on the type of organism, type of preservative, and the objective. And once they are all done, and we can have a most effective preservatives and also their optimal concentration, and we can formulate in the product and send it to the GMP laboratory to Amanda, and she can do some final testing, no matter for USP 51 or other regulation which need to follow.
Great. Well, thank you so much, Faye.
You're welcome.
Kind of a question regarding the final product, USP 51 testing. So if a product fails USP 51 testing, what should people be thinking about? What are the next steps there?
So this would depend on where the product is at. If it was a newly formulated product and it's coming in for the first time to be evaluated for USP 51 and there's failure, we would go back to the client, report the results, and likely they would need to do a reformulation. This could potentially mean that it's not the right preservative for the product, or maybe additional preservative needs to be added. And then if it was a product that was under stability testing and we were testing at, a time pull of, 18 months and the shelf life is, supposed to be 2 years. If you get a failure there, that's also indicative that, this preservative is not, it could be degrading and it's just not keeping the shelf life of the product. It's unable to inhibit the growth of the microorganisms. So at that point, the shelf life would either be changed or the client could opt to reformulate and use a different preservative.
Okay. Maybe as a follow-up, maybe considering USP 51 or other challenge studies, question come in regarding a beverage challenge study for growth of spoilage organisms. Can you conclude anything if you see growth during the first 10 days followed by a reduction or a leveling off by, say, the end or 28 days?
So, well, in terms of USP 51, what we would do is if we're testing at a 14-day and going to the 28 days, at 14 days, if we're seeing that the product is not meeting acceptance criteria, the client will be notified, because at that point they could opt to stop the testing. Most of the time we will still continue with the analysis because it's good information to have to see what would happen at that final 28 days.
Okay. Thank you. Faye, any considerations from your perspective on that?
Yeah, and for the beverage test, if it's not exactly needed to follow the USP 51, if it's like food beverage, also needed to consider about some FDA regulations. If it's so if it's not exactly following USP 51, so probably after day 14, you can consider other sampling time, like even day 21 or day 28, day 35, or depending on the spoilage organism, sometimes it's looking for the extended shelf life. So probably we can test it longer to see up to 1 month, 2 months, 3 months to see the organism can grow, because typically spoilage organism grows very slow. It's causing the spoilage, especially yeast and mold.
Makes sense. Okay, so shifting gears a little bit, I guess a clarification about USP 51 testing and how it applies. We talked a lot about personal care products, but not so much about food. Are there specific foods or categories within food that that method would apply for?
Yeah, for the food product, most of the regulation for the food product should be based on the FDA or USDA regulation. For example, like the acidified food products, that is we have the regulation for 21 CFR 114 for acidified food that is require 5 log reduction for pathogens like E. coli, Listeria, and Salmonella. If it's not acidified food, it's also need to follow some other regulation like FSMA and other CFR product like low acid food, 114, CFR 117, this type of thing. So based on different regulation, it has different requirement or recommendations about what the log reduction we should follow or the inhibition we should follow.
Makes sense. Amanda, any additions to that? Your thoughts?
So yeah, the USP 51 method is more specific towards personal care, pharmaceuticals. The products should be aqueous based, so we're ensuring that we're getting a homogeneous mixture of those microorganisms that we are adding. Now, while we couldn't say it complies with USP 51. We could do specialized studies following, a similar format that we would use for USP 51 testing. But again, we could provide the data, but we wouldn't be able to make a claim that it complies with the USP 51 method.
Okay. Perhaps a follow-up being for personal care, cosmetics, how would you decide between using USP method and other test methods then?
Well, for anything, any product that's regulated by a manufacturer that is registered with the FDA and needs to comply with CGMPs, the required test to show efficacy for that product would be USP 51.
Okay. Okay. Shifting over then, a couple questions on shelf life. So how does an inoculated pack challenge study relate to the shelf life of the food product? So if the shelf life of the product is 1 year, can we expect to carry on a challenge study for a year? Or what is the usual time frame for a challenge study to be conducted, and any considerations for accelerated shelf life studies?
That is a very good question. Yeah, for the shelf life study, we don't inoculate the product, but for the spoilage challenging study, we inoculate the spoilage organism into the product. So the difference is, if for the shelf life, we just save it for the shelf life, for example, for 1 year, or increasing the temperature and the humidity to do the accelerated shelf life. But also depending on if there is already some organism inside, so we don't know. But for the challenging study, we artificially inoculate the spoilage organism into the product, so we can make sure there are some organism inside, and to see if the product supports the growth of them. So maybe ideally we needed to test for the whole shelf life, but based on the real situation, we can do it shorter time, because if the organism can grow, it usually can happen in 1 or 2 months, because we inoculate relatively higher, around 3 logs, which is 1,000 CFU. Maybe in the real situation, it won't be that high in log concentration. So we challenge the system with a high inoculum level. So if the product support the organism to grow, it typically will happen earlier, like 1 or 2 months, or even 3 months. So it's depending on also depending on the timeline of the client. If they have plenty of time, we can do a longer time before the for the challenging study. But typically, if we do the spoilage challenging study, the spoilage can be showing up within 1 or 2 months.
Okay, thank you. I guess continuing on a little bit on challenge testing. So should a challenge testing on a food product be done on the factory manufactured product, or done on the R&D manufactured product? And if you kind of do one or the other, how do you bridge the differences, and where should you look for those differences?
Yeah, in R&D phase, we try to mimic the real situation in the manufacturer. For example, if the manufacturer are using thermal processing, and we try to in R&D phase, we also try to heat the product up to the temperature and a certain time to mimic the real situation happened in the pilot plant. And also when we design the R&D, for example, if the real product has a low pH, high acid, we always can adjust our model system or broth dilution in the R&D phase to also the similar pH. And also, if in the after the R&D phase done, and the client still find some want to use exactly like the manufacturer processing, we can also offer for the on-site validation. Like we formulate the formulated product with the preservative, and use surrogates to do the on-site validation to see the real manufacturer processing can also get the expected results compared to the R&D phase.
Very good, thank you. Let's go with one more question or two here. And this, I know our focus here is largely in microbiology. There's a question about the analytical stability of the preservative. Do your labs have familiarity with like maybe the chemical stability of the preservative, or what might you do if you needed to consider that with the client's needs here?
For the analytical stability of the preservatives, first we probably need to understand the principle of the preservatives. What the preservatives, how the preservatives working to inhibit the growth of the organism or inhibit or reduce. If it's relying on, for example, for like a vinegar, most of them relying on the acidity of the vinegar. So from analytical side, we will monitor the pH. And also for maybe some others, they are relying on the water activity of the product. We will also monitor the water activity. So it's more depending on what type of the final food product and also the principle how the preservatives inhibit the microorganisms.
Okay, thank you. I'm going to ask one more question. It's the last one that came in. Back on the challenge studies, are there any unique considerations for performing challenge study on a product after it's been opened, or after, you know, maybe you want to consider that following receipt, I suppose?
Yeah, that is also very typical inquiry we receive for the opened product, because during the manufacture or distribution, the temperature is relatively strictly controlled. For example, if it's a refrigeration condition, and the manufacturer always keep it under around 44 centigrade. But after it's open, probably the customer will leave it on the top for ambient temperature for a while, or the refrigeration temperature in their house is not strictly under 4 centigrade. So in this type of study for the opened product, we do some training study. Also, we want to see under abused temperature. Depending on the real storage condition is under the abused storage condition, if we still inoculate the organisms, and under this abused temperature, if it can grow or not.
Excellent.
So in our case here, while we don't typically receive products that are open to do a challenge study on, which we absolutely could, the point of doing the preservative testing using USP 51 is because most of these products, you know, your OTCs, cough syrups, you know, skincare, you know, they're multi-use. So we want to demonstrate that these preservatives are able to combat, you know, the consumer more or less contaminating the product, you know, possibly. So by doing that, you know, it shows that the preservative would be able to mitigate any of that growth that could potentially be put in the product by the consumer.
Makes sense.
Yeah, and most of the challenge study were other that we did is we open the product and artificially inoculate the product. So it's can be another way to say they are all open the product test.
That makes sense too. Okay, well, thank you. And I think that kind of wraps up our Q&A then. This is very interesting conversation here, seeing the start to finish essentially of the preservatives, and great perspectives from both of you during your talk today. So thank you both. Thank you, Faye. Thank you, Amanda. Thank you everybody for attending.
Thank you.
Thank you.